Welcome to Genomics and Java, a place for stories and conversations shared over our favorite cup. And this morning, we have Dr. Sara Cooper, one of our faculty investigators here at HudsonAlpha, is going to talk with us about the work that she does in her lab. So, Dr. Cooper, what is in your favorite cup this morning?
Well, I don't have my cup with me today, but I brought a Diet Coke, which is what I like to sip on when I'm sitting at my desk.
Excellent. So in your honor this morning, I am going to shift out and join you in in your favorite cup this morning. So tell us, what are some of your lab's current research interests?
So one of the biggest things that we're interested in right now is trying to understand how we can use genetic and genomic tools to understand how patients that have ovarian and pancreatic cancer specifically become resistant to some of the drugs that are commonly used to treat those cancers. So the work that we're doing uses some models, basically growing tumor cells in a dish, and allows us to test thousands of different genes all at the same time to determine whether they might play a role in drug resistance. And some of our recent work has shown that, yes, we can identify some of these genes. We've learned a little bit about what they do. And our long-term goal is to be able to actually use that information to predict how a patient who's diagnosed with one of these cancers might respond to a particular drug and hopefully identify a drug that would work the best for them.
That research is incredibly impactful. We know the statistics on cancer that so many of us are or have been impacted by cancer. So thank you so much for the work that you and your lab do. With all of the wonderful projects that you guys are working on, what would you consider your greatest contribution to science to date?
I have been thinking on that question, Jen, and I was having a hard time coming up with one specific thing, because one of the great things about the work that I've done here at HudsonAlpha and throughout my time in science is that I've been able to work on a variety of different projects. So we talked a lot today about the work that my lab does in cancer, which really is the primary focus of my lab right now. We've also done projects related to neuropsychiatric disease, which have been really interesting. That's just a small portion of some of the work that we've done, but it was a great way to sort of expand our horizons and think broadly about how the same tools that we're using in cancer can be relevant for other diseases too. And then, a little bit further back, I've done a lot of work in the more basic sciences, understanding things like how genes get turned on and turned off. So that's not an answer to your question, but I would say if I had to pick one thing, something that was really sort of transformative in my career was actually some of the early work I did really with the ENCODE project, which was part of that basic science work, trying to understand how genes get turned on and turned off. And one of the reasons I really enjoyed that is because it was a huge team effort, not only a team in the lab that I was working in, but also an international group of scientists. And I think that was really, got me thinking about how I want to do science as a team with other people. You kind of know I'm a people person, so I like to work with other people, talk with other people, hear what they're thinking, and come together to accomplish something. And so if I had to pick one thing that I was most sort of proud of, I think that would be it.
Sara, I think that is an amazing piece to bring to our audience, and to be able to talk about that science is such a process and that there's so much that's happening, and thinking about all of the teamwork and the collaboration and the opportunity to be a people person and to know that the work that you're doing every day is making a difference in the lives of both the collaborators that you work with as well as the people that you're working to solve problems for. And I think that's just such a beautiful part of science. Can you dive in a little bit more to a couple of the projects that your lab is working on? You mentioned being able to understand the genes that were turned on in some of the cancers becoming resistant to chemotherapy. Can you tell us a little bit more about some of those projects that you're working on?
Sure. So one project in particular is focused on pancreatic cancer. So that cancer, as you may know, is one of the most deadly cancers. Unfortunately, individuals that are diagnosed with pancreatic cancer really don't have a very good prognosis. The majority of individuals who are diagnosed with pancreatic cancer already have metastatic disease, meaning it has spread beyond the pancreas. And in those cases especially, it's very difficult to treat those cancers. Even for individuals that have earlier stage cancer at the time of diagnosis, they often still don't respond to the drugs that we have. And so one of the ideas is that we can use, instead of using what we call cytotoxic chemotherapies, which are just drugs that essentially kill all cells or impact all cells to some extent, but kill cancer cells more than healthy cells. Those are some of the best drugs that we have for treating pancreatic cancer, but as you might imagine, they have bad side effects because healthy cells are impacted as well. In some other cancers, the development of what we call targeted therapies has been really successful. That's the idea of identifying something that's specific to a tumor cell that you won't find in any healthy cells that you can use to find some way to target those specific cells in the body. And to date, we don't have any targeted therapies for pancreatic cancer, and so our work is really focused on why don't the cells that we have that are in pancreatic tumors respond very well to the therapies that we have, and can we identify new ways of treating those tumors with either a targeted therapy that might be more effective at killing the tumor cells rather than the healthy cells, and or could some of those targeted therapies be used in combination with existing therapies to improve how well they work, so that maybe you wouldn't have to have as many or as long of treatments, and that would, of course, make the side effects better and hopefully kill the tumors.
You have a very busy lab.
Yes, and that's just one of the projects, yeah.
What's another one of the projects that you guys are working on?
Yeah, so another really interesting project that we've done in collaboration with a physician at UAB, her name is Dr. Rebecca Arend, is focused on ovarian cancer. So ovarian cancer statistics are a little bit like those pancreatic cancer statistics that I talked about. The disease progresses a little bit differently. A lot of women who face ovarian cancer do respond to treatment initially, but then in most cases they recur sometimes a few months, sometimes a few years after that initial course of treatment. And so they ultimately end up facing some pretty poor survival outcomes because in the long run, the therapies that we have don't really work very well. And one of the ideas that's been out there to treat cancer in general and has been effective in some cancers is a treatment called immunotherapy. That's really a class of drugs that can be targeting lots of different things, but the basic idea is that they stimulate your own immune system to recognize tumor cells and kill them in the way that your body is supposed to, but that sometimes tumors are able to evade the immune system. And so immunotherapies have been tested in ovarian cancer, but they typically have not worked very well. There's a small number of patients that they'll work for, but in general, they're not very effective. And so our project with Dr. Rebecca Arend is really focused on understanding what are some of the factors that, first of all, might allow us to predict how a patient would respond to existing immunotherapies. And some of our more recent work, we've been able to identify some pathways, sets of genes that are important to how well the immune system is activated in ovarian cancer patients. And so our goal is to explore whether we can use new drugs to essentially inhibit those pathways that are preventing recognition of the tumors, with the goal being that we could use those drugs in combination with immunotherapies so that they would work better in hopefully all patients, or at least a greater portion of patients.
What a powerful set of tools to be able to put in the hands of our physicians and to make an impact on the lives of so many patients. You talked about being a people person, so what are some of your interests outside of the lab?
Well, I guess I have 2 kids, so mostly when I'm not in lab, I'm busy doing something with them. They like to play soccer, so on the weekends you'll find me on the soccer field with them. And as any good nerd, I guess, I like to read. So when I find time to do something on my own, reading is another thing. I like to watch movies with my husband.
I love it. What's one of your favorite books that you've read recently?
Oh my gosh, I haven't read anything really recently. I like The Handmaid's Tale. That's probably giving away too much, but...
Thank you so much for sharing a little bit about yourself and about the amazing work that your lab is doing. I'm going to close us out today with one last question. If you could solve one great mystery in genomics, what would it be, and why that particular mystery?
Okay, I have been thinking about this one. This is another tough question, Jen, but I think one of the things that I came up with that has been sort of a mystery in the field for quite some time is the idea that there's a lot of conditions that humans can be diagnosed with that we are pretty sure are genetic. When we say that, it's sort of like you might say runs in families. So mom has it, grandma has it, and over the last couple of decades, amazing work has been done to identify the genes and changes in our genome that are responsible for, those things running in families. So breast cancer is one example where there's a gene called BRCA1 that was identified, in the early 2000s now. So 20 years ago or so. That's been known for quite a while, and we can, if you see a family that has a lot of breast cancer in their family, you can sequence that gene and say, oh yeah, I think there's, that change right there, that increases their risk for breast cancer. But what we also see is families that look just as compelling where we can't find any changes in those genes that we know about already. And so there's sort of this bigger issue that's a good example of what's called missing heritability. So we know that there's things that are inherited through families, but we can't find the genetic changes that are responsible for them. And so where in the genome should we look? There's lots of great ideas about where it should be. So I guess that's a question that I would love to have the answer to, and maybe there's more than one answer. So it's a complex question, but probably has a complex answer too.
And I think that answer is one that resonates with so many of us as we look at families that we know and we see where they haven't had a genetics answer for that cancer that seems to run in their family. And so knowing that researchers like you and in your lab are continuing to work toward answering those questions, I think is an incredibly encouraging thing to focus on, especially at this time. So thank you, Sara, for joining us today and for talking about the work that you do and for the work that you and your lab do. And so until next time, stay curious and enjoy your favorite cup.